All right, this podcast is going to focus now, instead of on how we copy DNA, on how DNA actually builds this living tissue that is you. Okay, genes give the directions for making our physical bodies, okay? And telling our bodies how to function once they are made, okay? Now, before we go any further, hang on. What is a gene? Okay? Let's pretend that this is a long piece of DNA. We might also call this a chromosome. If you are a human, you have 46 of these in every single cell, chromosomes, okay? Now, what is a chromosome but a long piece of DNA? What is a piece of DNA except for A, T, C, G, A, T, 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 C, 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 a bunch of nucleotides strung together. A's, T's, C's, and G's all strung together in a particular order. Okay? Now, what is a gene? Well, let's say on this chromosome, we have AAT, GC, GG, GG, ATT, C, GC in this order going down that DNA strand. Okay? Well, those nucleotides in that order might be the genetic instructions for making melanin. Melanin is the pigment that gives your skin or your eyes or your hair color. Okay? And maybe that's those letters in that order, those nucleotides in that order, the instructions for making that pigment. But maybe right down from that, A, T, T, C, G, C, 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 maybe that is the instructions for making myosin. Myosin is the protein that makes your muscles contract. Maybe those are the instructions for that. That's another gene. Maybe down from that, there's A, C, 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 G, 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 T, T, A. Maybe that's the instructions for making hemoglobin, the protein in your blood. So a gene is nothing more than a sequence of letters on a chromosome, on a DNA strand. But Lord knows one DNA strand has thousands and thousands and thousands of letters, and so maybe just these certain letters here are for melanin. Maybe these letters here are for hemoglobin or whatever, okay? So that's what a gene is. And there can be many thousands of genes on one chromosome, okay? So, now, those, as we said, are the directions for building the different proteins that make up you, okay? So DNA is the directions to make the proteins, and then ultimately the proteins make up our cells, and the cells make up us. Okay? The DNA gets all the glory, but the proteins are what does all the work. Okay? And we are starting to learn more and more about proteins. For so many years, the focus was on DNA, but really we need to understand proteins to understand us. Now, please be aware that what we are focusing on in this podcast and the next one as well is this part of this puzzle right here, this getting from a DNA molecule to a protein molecule. This is called protein synthesis. Synthesis just means making, so protein making. Or it can also be called gene expression. When a gene expresses itself, it makes the protein that it is the directions for. So that's also called gene expression, so don't get confused by that statement. So, how do we get from a DNA molecule to a protein molecule? Okay? We already know how we get from DNA to DNA. We replicate it, and we've talked at length about how replication works, but replication is really not part of protein synthesis, and that you're just making DNA so that you can give DNA to your children, or if you're a skin cell, to your offspring skin cells. Okay? But what we want to talk about is how does DNA give the instructions for making this flesh and bone and blood that is us? And for that, we have to go through a bridge, an intermediary molecule called RNA, and then get to the proteins, which will then give us all of our traits that make us who we are. So transcription is the process where we go from DNA to our intermediary molecule RNA, and then later we will take that RNA and use that to make protein. But remember, DNA is just too important, it's too precious, it's the master plan, if you will, to be frivolously tossed around in the cell and out working on making proteins itself. It is protected and kept sequestered, while we can make many kind of disposable copies of RNA that help us make our proteins. Now, who figured out that genes, DNA molecules, are the instructions for making proteins? Who figured that out? Beadle and Tatum are the two scientists that figured out that genes code for proteins. Their idea at the time was one gene, one enzyme. Every gene in your DNA codes for an enzyme. But that's changed to one gene, one protein, because we know not all proteins are enzymes, and genes are the instructions for making all of our proteins, not just our enzymatic proteins. So it got modified to one gene, one protein. And it is further changing even today. In fact, scientists now believe that each gene area on a chromosome can actually code for several different proteins depending on how we splice those instructions together. So it's getting more complex as we understand more. All right, so what we're going to focus on now is transcription, which is this part right here going from DNA to RNA. We'll worry about this part later, not now. So we're going from DNA to RNA, okay? Now, DNA and RNA are really similar in all but three ways, okay? They're both nucleic acids made out of nucleotides and all that, but ribose sugar is used in RNA instead of deoxyribose, hence RNA instead of DNA. There is no thymine, there are no T's in RNA, so everywhere you would expect to see a T in an RNA molecule, you will actually see something called a U or a uracil nucleotide. It's just used in place of thymine. Okay? Also, RNA is single-stranded instead of being double-stranded. And what that means is RNA can kind of fold in complex ways sometimes that DNA can't, because it's all wound in this really pretty rigid, well, I don't want to say rigid, but that's the only word I've really got, molecule, whereas RNA can kind of bend and fold on itself rather protein-like. It's a little more, got a few more complex uses than DNA does. Also, there are lots of types of RNA, okay? There's mRNA, there's tRNA, and there's rRNA, and there's actually more than that, and we'll talk about what those mean in a second. All right, we talked about why have this RNA intermediate. It protects the DNA and doesn't make the DNA be the one have to go out wandering. We can keep the kind of DNA locked up in the safe because it's the blueprint, and we can send the RNA copies, and we can actually send many RNA copies of the same gene out into the world to make the protein that we need, so that we can get lots and lots and lots of the protein that we need. All right. So, we've got our DNA protected in the nucleus. We transcribe it and get many mRNA copies that can then be used to create a protein at the ribosome factories. Now, hopefully you remember this basic plan from your days in first year biology, okay? We'll be talking more about that as we go, however. So, how to make mRNA. It is really kind of similar to making DNA, but when we use the DNA as a template, just like we would when we replicate, but instead of transcribing both sides of that DNA helix, we only transcribe one of the original two DNA strands is transcribed. The one being transcribed actually is called the template. The other one that doesn't get any action is called the coding strand. So in this diagram down here, this part of the DNA here that is actually getting transcribed, and we are making the RNA by reading that DNA. This is the template strand. This up here would be the coding strand. And the synthesis of the RNA occurs at a opened up place in the DNA helix called a transcription bubble. Remember, we talked about a replication bubble. Well, now we have a transcription bubble. And the enzyme that is in charge, because remember, enzymes catalyze all these reactions, all these processes. The enzyme in charge is called RNA polymerase, very similar to DNA polymerase, but we are not making DNA, we are making RNA. And it is built in a 5' to 3' direction, just like DNA is. We can only add to the 3' end. All right, if you'll remember, I said there are several types of RNA. There is mRNA, that is actually messenger RNA. This is the important one. This is the one that actually carries the DNA genetic message out into the cytoplasm of the cell, where it's going to be used to make protein. tRNA is called transfer RNA, and we'll talk about what that does. rRNA we won't talk very much about, but that is called ribosomal RNA. And ribosomes are really important in making proteins, these little organelles, little tiny organelles that are out in the cytoplasm. And RNA is a component of what makes up a ribosome. A ribosome is made of protein and rRNA globbed together. It's not really a membrane-bound organelle like we've talked about chloroplast and mitochondria. It's just a glob of protein and RNA. And we won't talk about our RNA beyond that. There's also other kinds of RNA that we'll talk about later. Now, because there are several types of RNA, there are several types of enzymes that create it. RNA polymerase 1 makes rRNA, okay? RNA polymerase 2 is the one we're going to talk about, and that transcribes mRNA. RNA polymerase 3 makes tRNA. We don't really need to know more than just RNA polymerase makes RNA, okay? Now, there are 3 stages of making RNA, of transcription. There's starting it, initiation, doing it, elongation, and ending it, termination, okay? So, to start it, okay, we've got to know where to start. Remember, I said there's a gene for melanin on here, there's a gene for hemoglobin, there's a gene for myosin, there's a gene for growth hormone. So how do I know where to start? Where do I start with this? How do I know where's the beginning of the gene that I need? So, there's certain cues that the enzymes that do transcription look for. And these cues are called the promoter region. And these are regions on the DNA, so it's a sequence on the DNA that is found right before the start of the gene that you want. So if my myosin gene is here, say right here, then the promoter might be right above that, okay? And that's basically the part of the DNA that says start here for myosin, or whatever I just said this thing was. Okay? It tells the RNA polymerase to attach at that point to begin transcribing. Okay? The promoter sometimes is referred to as, yes, the Tata box. That's right, the Tata box, because there's lots of repetitions of TA, TA, and then AAA. So Tata box. Go figure. Okay? That is the signal for where the RNA polymerase and certain factors, certain proteins called transcription factors, are going to kind of attach to the DNA here and allow this transcription process to begin. Okay? So transcription factors, are just proteins that allow things to get started. Okay? I'm going to stop there for just a second, and I will catch you in a few minutes. Okay, so initiation, as you can see in this image, involves the transcription factors, which are just additional proteins that enable the RNA polymerase to bind to the promoter region on the DNA. Remember, the DNA is where the promoter sequence is that just tells the RNA polymerase and transcription factors, here's where we start transcribing, okay? Now, elongation, actually transcribing. All that's done here is that RNA polymerase actually matches the RNA bases to the DNA bases on that template strand. So down here we can tell this is DNA because we see T's and not U's. RNA polymerase just acts to add the complementary RNA nucleotides here. So if we see C, we know we're going to put a G. T, we're going to put A, but we see A here, we can't put a T, so we're going to put a uracil, and so on. Okay, and remember we are going in a 5' to 3' direction, just like we have to do in replication. Now, termination, ending it, stopping transcription. We hit a terminator sequence, which is something like AAUAAA on DNA. On the DNA, well, this would have to be a little bit different. This is kind of a typo here, my bad, because it's not going to be a U on the DNA, so it would have to be complementary to this. But actually, you really don't need to know this. Just understand you hit a terminator sequence of nucleotides that tells RNA polymerase to stop, and so it does. The transcript that was just made is now ready to not go make a protein yet, but to get a little sliced and diced and finally prepared through something called RNA processing, and then it will be ready to help us make a protein. So what we have to talk about next is RNA processing, and I will catch you in the next podcast for that. Thanks for watching. See you soon.